A few years ago, with my colleague Emmanuel Charpentier, I invented a new technology for editing genomes. It's called CRISPR-Cas9. The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. You might be interested to know that the CRISPR technology came about through a basic research project that was aimed at discovering how bacteria fight viral infections. Bacteria have to deal with viruses in their environment, and we can think about a viral infection like a ticking time bomb. A bacterium has only a few minutes to defuse the bomb before it gets destroyed. So many bacteria have in their cells an adaptive immune system called CRISPR that allows them to detect viral DNA and destroy it. Part of the CRISPR system is a protein called Cas9 that's able to seek out and cut and eventually degrade viral DNA in a specific way. And it was through our research to understand the activity of this protein Cas9 that we realized that we could harness its function as a genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed recently that they could even use the CRISPR technology to change genes in human embryos. And scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. The opportunity to do this kind of genome editing also raises various ethical issues that we have to consider, because this technology can be employed not only in adult cells, but also in the embryos of organisms, including our own species. And so together with my colleagues, I've called for a global conversation about the technology that I co-invented so that we can consider all of the ethical and societal implications of a technology like this. What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection. And as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories, or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things, like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues. Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented littermates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s, when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough. Thank you.
Jennifer, this is a technology with huge consequences. You pointed out your attitude about asking, you can call it a pause or a moratorium or a quarantine, is incredibly responsible. There are, of course, the therapeutic results of this, but then there are the non-therapeutic ones, and they seem to be the ones gaining traction, particularly in the media. This is one of the latest issues of The Economist. Editing humanity is all about genetic enhancement, it's not about therapeutics, right? What kind of reactions did you get back in March from your colleagues in the science world when you asked or when you suggested that we should actually pause this for a moment and think about it?
My colleagues were actually, I think, delighted to have the opportunity to discuss this openly. It's interesting that as I talk to people, my scientific colleagues as well as others, there's a wide variety of viewpoints about this. So clearly it's a topic that needs careful consideration and discussion.
So there's a big meeting happening in December that
Correct.
You and your colleagues are calling together with the Academy of Science and others. What is your expectation? What do you hope that will come out of the meeting practically?
Well, I hope that we can air the views of many different individuals and stakeholders who want to think about how to use this technology responsibly. It may not be possible to come up with a consensus point of view, but I think we should at least understand what all the issues are as we go forward.
Now, colleagues of yours, like George Church, for example, at Harvard, they say, yeah, ethical issues basically are just a question of safety. We test and test and test again in animals and in labs, and then once we feel it's safe enough, we move on to humans. And so that's kind of the other school of thought, is we should actually use this opportunity and really go for it. Is there a possible split happening in the science community about this? I mean, are we going to see some people holding back because they have ethical concerns and some others just going for it because some countries under-regulate or don't regulate at all?
Well, I think with any new technology, especially something like this, there are going to be a variety of viewpoints. And I think that's perfectly understandable. I think that in the end, this technology will be used for human genome engineering. But I think that to do that without careful consideration and discussion of the risks and the potential complications would not be responsible.
Now, there are a lot of technologies in other fields of science that are developing exponentially, pretty much like yours. I'm thinking about artificial intelligence, about autonomous robots and so, no one seems, aside from autonomous warfare robots, nobody seems to have launched a similar discussion in those fields and calling for a moratorium. Do you think that your discussion may serve as a blueprint for other fields?
Well, I think it's hard for scientists to get out of the laboratory. Speaking for myself, it's a little bit uncomfortable to do that. But I do think that being involved in the genesis of this really puts me and my colleagues in a position of responsibility. And I would say that I certainly hope that other technologies will be considered in the same way, just as we would want to consider something that could have implications in other fields besides biology.
Jennifer, thanks for coming to TED. Thank you very much.
Thank you.
So has everybody heard of CRISPR? I would be shocked if you hadn't. This is a technology, it's for genome editing, and it's so versatile and so controversial that it's sparking all sorts of really interesting conversations. Should we bring back the woolly mammoth? Should we edit a human embryo? And my personal favorite, how can we justify wiping out an entire species that we consider harmful to humans off the face of the earth using this technology? This type of science is moving much faster than the regulatory mechanisms that govern it. And so for the past 6 years, I've made it my personal mission to make sure that as many people as possible understand these types of technologies and their implications. Now, CRISPR has been the subject of just a huge media hype. And the words that are used most often are easy and cheap. So what I want to do is drill down a little bit deeper and look into some of the myths and the realities around CRISPR. If you're trying to CRISPR a genome, the first thing that you have to do is damage the DNA. The damage comes in the form of a double-strand break through the double helix, and then the cellular repair processes kick in, and then we convince those repair processes to make the edit that we want and not a natural edit. That's how it works. It's a two-part system. You've got a Cas9 protein and something called a guide RNA. I like to think of it as a guided missile. So the Cas9, I love to anthropomorphize. So the Cas9 is kind of this Pac-Man thing that wants to chew DNA. And the guide RNA is the leash that's keeping it out of the genome until it finds the exact spot where it matches. And the combination of those two is called CRISPR. It's a system that we stole from an ancient bacterial immune system. The part that's amazing about it is that the guide RNA, only 20 letters of it are what target this system. This is really easy to design, and it's really cheap to buy. So that's the part that is modular in the system. Everything else stays the same. This makes it a remarkably easy and powerful system to use. So the guide RNA and the Cas9 protein, complex together, go bouncing along the genome. And when they find a spot where the guide RNA matches, then it inserts between the two strands of the double helix, it rips them apart, that triggers the Cas9 protein to cut, and all of a sudden, you've got a cell that's in total panic because now it's got a piece of DNA that's broken. What does it do? It calls its first responders. There are two major repair pathways. The first just takes the DNA and shoves the two pieces back together. This isn't a very efficient system because what happens is sometimes a base drops out or a base is added. It's an okay way to maybe knock out a gene, but it's not the way that we really want to do genome editing. The second repair pathway is a lot more interesting. In this repair pathway, it takes a homologous piece of DNA, and now, mind you, in a diploid organism like people, we've got one copy of our genome from our mom and one from our dad. So if one gets damaged, it can use the other chromosome to repair it. So that's where this comes from. The repair is made, and now the genome is safe again. The way that we can hijack this is we can feed it a false piece of DNA, a piece that has homology on both ends but it's different in the middle. So now you can put whatever you want in the center and the cell gets fooled. So you can change a letter, you can take letters out, but most importantly, you can stuff new DNA in, kind of like a Trojan horse. CRISPR is going to be amazing in terms of the number of different scientific advances that it's going to catalyze. The thing that's special about it is this modular targeting system. I mean, we've been shoving DNA into organisms for years, right? But because of the modular targeting system, we can actually put it exactly where we want it. The thing is that there's a lot of talk about it being cheap and it being easy, and I run a community lab. I'm starting to get emails from people that say stuff like, hey, can I come to your open night and maybe use CRISPR and engineer my genome? Seriously. No, you can't. But I've heard it's cheap, I've heard it's easy. So we're going to explore that a little bit. So how cheap is it? Yeah, it is cheap in comparison. It's going to take the cost of the average materials for an experiment from thousands of dollars to hundreds of dollars, and it cuts the time a lot too. It can cut it from weeks to days. That's great. You still need a professional lab to do the work in. You're not going to do anything meaningful outside of a professional lab. I mean, don't listen to anyone who says you can do this sort of stuff on your kitchen table. It's really not easy to do this kind of work. Not to mention, there's a patent battle going on. So even if you do invent something, the Broad Institute and the UC Berkeley are in this incredible patent battle. It's really fascinating to watch it happen because they're accusing each other of fraudulent claims, and then they've got people saying, oh, I signed my notebook here or there. This isn't going to be settled for years. And when it does, you can bet you're going to pay someone a really hefty licensing fee in order to use this stuff. So is it really cheap? Well, it's cheap if you're doing basic research and you've got a lab. So how about easy? Let's look at that claim. The devil is always in the details. We don't really know that much about cells. They're still kind of black boxes. So for example, we don't know why some guide RNAs work really well and some guide RNAs don't. We don't know why some cells want to do one repair pathway and some cells would rather do the other. And besides that, there's the whole problem of getting the system into the cell in the first place. In a petri dish, that's not that hard, but if you're trying to do it on a whole organism, it gets really tricky. It's okay if you use something like blood or bone marrow. Those are the targets of a lot of research now. There was a great story of some little girl who they saved from leukemia by taking the blood out, editing it, putting it back with a precursor of CRISPR. And this is a line of research that people are going to do. But right now, if you want to get into the whole body, you're probably going to have to use a virus. So you take the virus, you put the CRISPR into it, you let the virus infect the cell. But now you've got this virus in there. And we don't know what the long-term effects of that are. Plus, CRISPR has some off-target effects, very small percentage, but they're still there. So what's going to happen over time with that? These are not trivial questions. And there are scientists that are trying to solve them, and they will eventually, hopefully, be solved. But it ain't plug and play, not by a long shot. So is it really easy? Well, if you spend a few years working it out in your particular system, Yes, it is. Now, the other thing is we don't really know that much about how to make a particular thing happen by changing particular spots in the genome. We're a long way away from figuring out how to give a pig wings, for example. Or even an extra leg. I'd settle for an extra leg. That would be kind of cool, right? But what is happening is that CRISPR is being used by thousands and thousands of scientists to do really, really important work, like making better models of diseases in animals, for example, or for taking pathways that produce valuable chemicals and getting them into industrial production and fermentation vats, or even doing really basic research on what genes do. This is the story of CRISPR we should be telling, and I don't like it that the flashier aspects of it are drowning all of this out. So I would urge you to really learn about these types of technologies, because really only in that way are we going to be able to guide the development of these technologies, the use of these technologies, and make sure that in the end, it's a positive outcome for both the planet and for us. Thanks.
I'd like to introduce you to Laika. To most of us, Laika is simply a very cute pig. However, to hundreds of thousands of patients in need of a life-saving organ, Laika is a symbol of hope. You see, ever since the 1970s, when organ transplants became a real option for patients with kidney failure and other organ diseases, organ supply has been an issue. Over the last few decades, the issue only worsened as organ demand has exponentially increased. Currently, in the US, there are close to 115,000 patients in need of life-saving organ transplant. By the end of my talk, one more patient will be added to this list. Today, about 100 people will get a new organ, a chance to start their life anew, and yet, by the end of today, 20 others will die waiting. The situation is heartbreaking for patients, for their families, and for the doctors who want to do more. In some part of the world, the situation also became a disturbing social issue. In Asia, for example, media outlets reported that desperate patients are obtaining organs from the cruel black market. It is clear that a solution is needed to this crisis. Human lives are at stake. As a biologist and a geneticist, it has become my mission to help solve this problem. Today, I'm optimistic to say that we are on our way there, thanks to Laika. Using gene editing technology, it's now possible to exquisitely create human transplantable organ that can be safely grown in pigs. Before we jump into the incredible science that makes it happen, let's have a better understanding what xenotransplantation is. It's a process of transplanting animal organs into humans. You may want to ask, why pig organs? Because some pigs carry organs with similar size and physiology to human organs. Over the last half a century, pioneers of transplantation have tried hard to make it happen, but with limited to no success. Why is that? Two fundamental hurdles stood in the way. First is the problem of rejection. When our immune system sees a new organ as foreign, it will reject it. Second, and this one is specific to the organs from the pig. Every pig carries a virus that is benign to the pig but can be transmitted into human. It is called the porcine endogenous retrovirus, PERV. And this virus has the potential to cause a viral epidemic similar to HIV. Without an effective way to address these issues, the field of xenotransplantation has been on hold for more than 1 decade. Little progress has been made until now. Let me share with you how I got here today with Leica. My journey started from Emei Mountain in China. That is a place well described in a lot of legendary stories, like the crouching tiger, hidden dragon. That is the place I call home. Growing up in the mountain, I started to have strong connection with nature. This is me when I was 7 years old, standing in front of an ancient Buddhist temple with a monkey on my shoulder. I still vividly remember how my friends and I would toss the peanuts around to distract the monkeys so that we can cross to hike through the valley. I love nature. When it was time to choose a field of study, I chose to study biology at Peking University in Beijing. However, the more I learned, the more questions I had. How could our genetic makeup be so similar to animals and yet we look so different? How is our immune system capable of fighting off so many pathogens but smart enough not to attack ourselves? Questions like this tormented me. I know it sounds nerdy, but you know I'm a scientist. After college, I decided I didn't want to just ask the questions. I wanted to answer them, so I did. In 2008, I was lucky enough to be accepted into the PhD program at Harvard University and worked with Dr. George Church. While working in Church's lab, I started to learn and experiment the genetic makeup of mammals. Among all the experiments, one particular one took me closer to Leica. In 2013, my colleagues and I made changes in a human cell using a tool you may have heard about called Crispr. We were one of the first 2 groups to report the successful use of such a tool in changing our DNA. It was an exciting moment in scientific discovery. The gene editing tool Crispr has 2 components. It has a scissor called the enzyme Crispr and what is called a guide RNA. Think about it as a genetic scissors with a microscope. The microscope is a guide RNA, which brings the scissors to the place we want to cut. And it says, here it is, and the enzyme Crispr just cut and repair the DNA in the way we want. Shortly after we reported our study, physicians at Mass General Hospital were intrigued by the medical applications of our research. They reached out to us, and together we began to see the potential to use Crispr to solve the organ shortage crisis. How do we do it? It is simple, yet very complex. We started by making changes in a pig cell to make it virus-free and human immune compatible. The nucleus of that cell is then implanted into a pig egg and allowed to divide into an embryo. The resulting embryo is then placed into the uterus of a surrogate mother and allowed to divide into a pig. Basically, it's a process of cloning. The piglet then carries organs whose genetic makeup hopefully wouldn't be rejected by the human immune system. In 2015, our team decided to tackle the viral transmission problem first. We wanted to take out all 62 copies of the perf virus from the pig genome. By that time, it was nearly mission impossible. Even with CRISPR, we could only do 1 or 2 modifications within a cell. The record for number of modifications we can do in a particular cell was 5. We had to increase the throughput by more than 10-folds to achieve that. With very careful design and hundreds of trials, we successfully took out all the virus, broke the record. More importantly, our study showed that we could eliminate the possibility of this dangerous virus being transmitted into humans. Last year, with a modified cell and cloning technology, our startup, eGenesis, produced Leica, the first pig of its kind born without perf. Leica represents the first critical step in establishing the safe xenotransplantation. It is also a platform that we can do further genetic modification on to solve the immunology problem. Since then, we have created more than 30 pigs without perf, and they may be the most advanced genome-modified animal living on Earth. We named Leica after the Soviet dog, who was the first animal to orbit the Earth. We hope Leica and her siblings can lead us into a new frontier of science and medicine. Imagine a world where patients suffer from liver failure can be saved with a new liver without having to wait for a donation or another human to die. Imagine a world where people with diabetes do not have to rely on insulin after every meal because we can provide them with good pancreatic cells that can produce insulin on their own. And imagine a world where patients with kidney failure do not have to face the burden of dialysis. We are striving to create that world, a world without organ shortage. We finally have the tool to tackle the problem we could never tackle before, and Leica is just the beginning of our journey. We have to be very humble in front of the nature because there are more issues to be addressed, including immunology and things we couldn't even anticipate at this point. However, it is our responsibility to translate the cutting-edge science into medicine to save the lives of all the patients who are waiting. Thank you very much.
I mean, Luhan, this is extraordinary work. Here, come forward. So, what's the next steps here? You've got rid of the virus. The next steps involve trying to get to the point where a human body won't reject a transplant. What's involved in solving that?
It's a very complicated process. So we need to take out the antigen of the pigs. In addition, we can learn a lot from cancer. How can cancer invade or circumvent our immune system so that we can utilize the trick of cancer and implement that on the pig organ to fool our immune system to not attack the organ.
When would you estimate, when do you hope that the first successful transplant would happen?
It will be irresponsible for me to give you any number.
We're at TED, we're always irresponsible.
But we are working day and night trying to make this happen for the patients.
So not even, you won't say that you think it could happen within a decade or within 5 years or something? Or
for sure, we hope it happens within 1 decade.
So there's a lot of people here who'll be very, very excited at that, and I mean, the potential for this is extraordinary. There'll be some other people here who are going, that pig is too cute. Humans shouldn't be exploiting something so cute for our benefit. Do you have any response to that?
Yeah, sure. So imagine one pig can save 8 people's life. In addition, similar to human donation, if we only harvest 1 kidney from the pig can still alive. So we are very mindful about the issues, but I think our goal is just to address the unmet medical need for those patients and their families.
Plus, no one can say that to you if they eat bacon, right?
That's a good point.
Johan, thank you so much.
Thank you so much.
So this is a talk about gene drives. I'm going to start by telling you a brief story. 20 years ago, a biologist named Anthony James got obsessed with the idea of making mosquitoes that didn't transmit malaria. It was a great idea and pretty much a complete failure. For one thing, it turned out to be really hard to make a malaria-resistant mosquito. James managed it finally, just a few years ago, by adding some genes that make it impossible for the malaria parasite to survive inside the mosquito. But that just created another problem. Now that you've got a malaria-resistant mosquito, how do you get it to replace all the malaria-carrying mosquitoes? There were a couple options, but plan A was basically to breed up a bunch of the new genetically engineered mosquitoes, release them into the wild, and hope that they pass on their genes. The problem was that you'd have to release literally 10 times the number of native mosquitoes for that to work. So in a village with 10,000 mosquitoes, you'd release an extra 100,000. As you might guess, this was not a very popular strategy with the villagers. Then, last January, Anthony James got an email from a biologist named Ethan Beer. Beer said that he and his grad student, Valentino Gantz, had stumbled on a tool that could not only guarantee that a particular genetic trait would be inherited, but that would spread incredibly quickly. If they were right, it would basically solve the problem that James had been working on for 20 years. As a test, they engineered 2 mosquitoes to carry the anti-malaria gene and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the baby should come out in 4 permutations: AA, AB, AA, BA. Instead, with a new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as Crispr in 2012. Many of you have probably heard about Crispr, so I'll just say briefly that Crispr is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had 2 problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to Crispr. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that Crispr inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if Crispr also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This Crispr gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace. Or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool. Up until now, the fact that gene drives didn't work very well was actually kind of a relief. Normally, when we mess around with an organism's genes, we make that thing less evolutionarily fit. So biologists can make all the mutant fruit flies they want without worrying about it. If some escape, natural selection just takes care of them. What's remarkable and powerful and frightening about gene drives is that that will no longer be true. Assuming that your trait does not have a big evolutionary handicap, like a mosquito that can't fly, the CRISPR-based gene drive will spread the change relentlessly until it is in every single individual in the population. Now, it isn't easy to make a gene drive that works that well, but James and Esvelt think that we can. The good news is that this opens the door to some remarkable things. If you put an anti-malarial gene drive in just 1% of Anopheles mosquitoes, the species that transmits malaria, researchers estimate that it would spread to the entire population in a year. So in a year, you could virtually eliminate malaria. In practice, we're still a few years out from being able to do that, but still, 1,000 children a day die of malaria. In a year, that number could be almost 0. The same goes for dengue fever, chikungunya, yellow fever. And it gets better. Say you want to get rid of an invasive species, like get Asian carp out of the Great Lakes. All you have to do is release a gene drive that makes the fish produce only male offspring. In a few generations, there'll be no females left. No more carp. In theory, this means we could restore hundreds of native species that have been pushed to the brink. Okay, that's the good news. This is the bad news. Gene drives are so effective that even an accidental release could change an entire species, and often very quickly. Anthony James took good precautions. He bred his mosquitoes in a biocontainment lab, and he also used a species that's not native to the US, so that even if some did escape, they'd just die off. There'd be nothing for them to mate with. But it's also true that if a dozen Asian carp with the all-male gene drive accidentally got carried from the Great Lakes back to Asia, they could potentially wipe out the native Asian carp population. And that's not so unlikely, given how connected our world is. In fact, it's why we have an invasive species problem. And that's fish. Things like mosquitoes and fruit flies, there's literally no way to contain them. They cross borders and oceans all the time. Okay, the other piece of bad news is that a gene drive might not stay confined to what we call the target species. That's because of gene flow, which is basically a fancy way of saying that neighboring species sometimes interbreed. If that happens, it's possible a gene drive could cross over, like Asian carp could infect some other kind of carp. That's not so bad if your drive just promotes a trait, like eye color. In fact, there's a decent chance that we'll see a wave of very weird fruit flies in the near future. But it could be a disaster if your drive is designed to eliminate the species entirely. The last worrisome thing is that the technology to do this, to genetically engineer an organism and include a gene drive, is something that basically any lab in the world can do. An undergraduate can do it. A talented high schooler with some equipment can do it. Now, I'm guessing that this sounds terrifying. Interestingly, though, nearly every scientist I talked to seemed to think that gene drives were not actually that frightening or dangerous. Partly that's because they believe that scientists will be very cautious and responsible about using them. So far, that's been true. But gene drives also have some actual limitations. So for one thing, they work only in sexually reproducing species, so thank goodness, they can't be used to engineer viruses or bacteria. Also, the trait spreads only with each successive generation. So changing or eliminating a population is practical only if that species has a fast reproductive cycle, like insects or maybe small vertebrates like mice and fish. In elephants or people, it would take centuries for a trait to spread widely enough to matter. Also, even with CRISPR, it's not that easy to engineer a truly devastating trait. Say you wanted to make a fruit fly that feeds on ordinary fruit instead of rotting fruit with the aim of sabotaging American agriculture. First, you'd have to figure out which genes control what the fruit fly wants to eat, which is already a very long and complicated project. Then you'd have to alter those genes to change the fly's behavior to wherever you want it to be, which is an even longer and more complicated project. And it might not even work because the genes that control behavior are complex. So if you're a terrorist and have to choose between starting a grueling basic research program that will require years of meticulous lab work and still might not pan out, or just blowing stuff up, you'll probably choose the latter. This is especially true because, at least in theory, it should be pretty easy to build what's called a reversal drive. That's one that basically overwrites the change made by the first gene drive. So if you don't like the effects of a change, you can just release a second drive that will cancel it out, at least in theory. Okay, so where does this leave us? We now have the ability to change entire species at will. Should we? Are we gods now? I'm not sure I'd say that, but I would say this. First, some very smart people are even now debating how to regulate gene drives. At the same time, some other very smart people are working hard to create safeguards, like gene drives that self-regulate or peter out after a few generations. That's great. But this technology still requires a conversation. And given the nature of gene drives, that conversation has to be global. What if Kenya wants to use a drive, but Tanzania doesn't? Who decides whether to release a gene drive that can fly? I don't have the answer to that question. All we can do going forward, I think, is talk honestly about the risks and benefits, and take responsibility for our choices. By that I mean not just the choice to use a gene drive, but also the choice not to use one. Humans have a tendency to assume that the safest option is to preserve the status quo. But that's not always the case. Gene drives have risks, and those need to be discussed. But malaria...
So what if I could make for you a designer baby? What if you as a parent-to-be and I as a scientist decided to go down that road together? What if we didn't? What if we thought, we thought that's a bad idea? But many of our family, friends, and coworkers did make that decision. Let's fast forward just 15 years from now. Let's pretend it's the year 2030, and you're a parent. You have your daughter, Marion, next to you. And in 2030, she is what we call a natural because she has no genetic modifications. And because you and your partner consciously made that decision, many in your social circle, they kind of look down on you. They think you're like a Luddite or a technophobe. Marion's best friend, Jenna, who lives right next door, is a very different story. She was born a genetically modified designer baby with numerous upgrades. Yeah, upgrades. And these enhancements were introduced using a new genetic modification technology that goes by the funny name Crispr. Like something's crisp. This is Crispr. The scientists that Jenna's parents hired to do this for several $1 million introduced Crispr into a whole panel of human embryos. And then they used genetic testing, and they predicted that that little tiny embryo, Jenna's embryo, would be the best of the bunch. And now, Jenna is an actual real person. She's sitting on the carpet in your living room, playing with your daughter, Marion. And your families have known each other for years now. And it's become very clear to you that Jenna is extraordinary. She's incredibly intelligent. If you're honest with yourself, she's smarter than you. And she's 5 years old. She's beautiful, tall, athletic, and the list goes on and on. And in fact, there's a whole new generation of these GM kids like Jenna. And so far, it looks like they're healthier than their parents' generation, than your generation. And they have lower healthcare costs. They're immune to a host of health conditions, including HIV/AIDS and genetic diseases. It all sounds so great. But you can't help but have this sort of unsettling feeling, a gut feeling, that there's something just not quite right about Jenna. And you've had the same feeling about other GM kids that you've met. You were also reading in the newspaper earlier this week that a study of these children who were born as designer babies indicates they may have some issues like increased aggressiveness and narcissism. But more immediately on your mind is some news that you just got from Jenna's family. She's so smart. She's now going to be going to a special school, a different school than your daughter, Marion. And this has kind of thrown your family into a disarray. Marion's been crying, and last night, when you took her to bed to kiss her goodnight, she said, "Daddy, will Jenna even be my friend anymore?" So now, as I've been telling you this Imagine 2030 story, I have a feeling that I may have put some of you into this like sci-fi frame of reference, right? You think you're reading a sci-fi book, or maybe like in Halloween, mode of thinking. But this is really a possible reality for us just 15 years from now. I'm a stem cell and genetics researcher, and I can see this new CRISPR technology and its potential impact. And we may find ourselves in that reality. And a lot will depend on what we decide to do today. And if you're still kind of thinking in sci-fi mode, consider that the world of science had a huge shock earlier this year, and the public largely doesn't even know about it. Researchers in China just a few months ago reported the creation of genetically modified human embryos. This was the first time in history. And they did it using this new CRISPR technology. It didn't work perfectly, but I still think they've sort of cracked the door ajar on a Pandora's box here. And I think some people are going to run with this technology and try to make designer babies. Now, before I go on, some of you may sort of hold up your hands and say, stop, Paul, wait a minute, wouldn't that be illegal, right? You can't just go off and create a designer baby. And in fact, to some extent, you're right. In some countries, you couldn't do that. But in many other countries, including my country, the US, there's actually no law on this. So in theory, you could do it. And there was another development this year that resonates in this area. And that happened not so far from here, over in the UK. And the UK traditionally has been the strictest country when it comes to human genetic modification. It was illegal there. But just a few months ago, they carved out an exception to that rule. They passed a new law allowing the creation of genetically modified humans with the noble goal of trying to prevent a rare kind of genetic disease. But still, I think, in combination, these events are pushing us further towards sort of an acceptance of human genetic modification. So I've been talking about this CRISPR technology. What actually is CRISPR? So if you think about the GMOs that we're all more familiar with, like GMO tomatoes and wheat and things like that, this technology is similar to the technologies that were used to make those, but it's dramatically better, cheaper, and faster. So what is it? It's actually like a genetic Swiss Army knife. We can pretend this is a Swiss Army knife with different tools in it. And one of the tools is kind of like a magnifying glass or a GPS for our DNA. So it can home in on a certain spot. And the next tool is like a scissors that can cut the DNA right in that spot. And finally, we have a pen where we can literally rewrite the genetic code in that location. It's really that simple. And this technology, which came on the scene just 3 years ago, has taken science by storm. It's evolving so fast, and it's so freaking exciting to scientists. And I admit, I'm fascinated by it, and we use it in my own lab, that I think someone's going to go that extra step and continue the GM human embryo work and maybe make designer babies. This is so ubiquitous now. It just came on the scene 3 years ago. Thousands of labs literally have this in hand today. And they're doing important research. Most of them are not interested in designer babies. They're studying human disease and other important elements of science. So there's a lot of good research going on with CRISPR. And the fact that we can now do genetic modifications that used to take years and cost millions of dollars in a few weeks for a couple $1,000, to me as a scientist, that's fantastic. But again, at the same time, it opens the door to people going too far. And I think for some people, the focus is not going to be so much on science. That's not what's going to be driving them. It's going to be ideology or a chase for a profit. And they're going to go for designer babies. So why should we be, why be concerned about this? We know from Darwin, if we go back 2 centuries, the evolution and genetics profoundly have impacted humanity, who we are today. And some think there's like a social Darwinism at work in our world, and maybe even a eugenics as well. Imagine those trends, those forces, with a booster rocket of this CRISPR technology that is so powerful and so ubiquitous. And in fact, we can just go back 1 century to the last century to see the power that eugenics can have. So my father, Peter Knopfler, was actually born right here in Vienna. He was Viennese. And he was born here in 1929. And when my grandparents had little baby Peter, the world was very different, right? It was a different Vienna. The United States was different. The world was different. There was a eugenics rising. And my grandparents realized pretty quickly, I think, that they were on the wrong side of the eugenics equation. And so despite this being their home and their whole extended family's home, and this area, being their family's home for generations, they decided, because of eugenics, that they had to leave. And they survived, but they were heartbroken. And I'm not sure my dad ever really got over leaving Vienna. He left when he was just 8 years old in 1938. So today, I see a new eugenics kind of bubbling to the surface. It's supposed to be a kinder, gentler, positive eugenics, different than all that past stuff. But I think even though it's focused on trying to improve people, it could have negative consequences. And it really worries me that some of the top proponents of this new eugenics, they think CRISPR is the ticket to make it happen. So I have to admit, eugenics, we talk about making better people. It's a tough question, what is better when we're talking about a human being? But I admit, I think maybe a lot of us could agree that, human beings, maybe we could use a little betterment. Look at our politicians. Here, back in the US, God forbid we go there right now. Maybe even if we just look in the mirror, there might be ways we think, we could be better. I might wish, honestly, that I had more hair here, instead of baldness. Some people might wish they were taller, have a different weight, a different face. If we could do those things, we could make those things happen, or we could make them happen in our children, it would be very seductive. And yet, coming with it would be these risks. I talked about eugenics, but there would be risks to individuals as well. So if we forget about enhancing people and we just try to make them healthier using genetic modification, this technology is so new and so powerful that by accident we could make them sicker. That easily could happen. And there's another risk, and that is that all of the legitimate, important genetic modification research going on just in the lab, again, no interest in designer babies, a few people going the designer baby route, things go badly, that entire field could be damaged. I also think it's not that unlikely that governments might start taking an interest in genetic modification. So, for example, our imagined GM Jenna child who is healthier, if there's a generation that looks like they have lower healthcare costs, it's possible that governments may start trying to compel their citizens to go the GM route. Look at China's one-child policy. It's thought that that prevented the birth of 400 million human beings. So it's not, beyond the realm of possible that genetic modification could be something that governments push. And if designer babies become popular, in our digital age, viral videos, social media, what if designer babies are thought to be fashionable and they kind of become the new glitterati, right? The new Kardashians or something. Are those trends that we really could control? I'm not convinced that we could. So again, today it's Halloween, and when we talk about genetic modification, there's one Halloween-associated character that is talked about or invoked more than anything else, and that is Frankenstein, right? Mostly that's been, Franken foods and all this other stuff. But if we think about this now, and we think about it in the human context, on a day like Halloween, if parents can, in essence, costume their children genetically, are we going to be talking about a Frankenstein 2.0 kind of situation? I don't think so. I don't think it's going to get to that extreme. But when we are going about hacking the human code, I think all bets are off in terms of what might come of that. There would still be dangers. And we can look in the past to other elements of transformative science and see how they can basically go out of control and permeate society. So I'll just give you one example, and that is in vitro fertilization. Almost exactly 40 years ago, test tube baby number 1, Louise Brown, was born. And that's a great thing. And I think since then, 5 million IVF babies have been born, bringing immeasurable happiness. A lot of parents now can love those kids. But if you think about it, in 4 decades, 5 million babies being born from a new technology is pretty remarkable. And the same kind of thing could happen with human genetic modification and designer babies. So, depending on the decisions we make in the next few months, the next year or so, if designer baby number 1 is born, within a few decades, there could well be millions of genetically modified humans. And there's a difference there, too, because if we you in the audience or I, if we decide to have a designer baby, then their children will also be genetically modified, and so on, because it's heritable. So that's a big difference. So with all of this in mind, what should we do? There's actually going to be a meeting a month from tomorrow in Washington, D.C., by the U.S. National Academy of Sciences to tackle that exact question. What is the right path forward with human genetic modification? I believe at this time we need a moratorium. We have to ban this. We should not allow creating genetically modified people because it's just too dangerous and too unpredictable. But there's a lot of people. Thanks. And let me say, just as a scientist, it's a little bit scary for me to say that in public because science, generally doesn't like self-regulation and things like that. So I think, we need to put a hold on this. But there are many people who not only disagree with me, they feel the exact opposite. They're like, step on the gas, full speed ahead, let's make designer babies. And so in the meeting in December and other meetings that are likely to follow in the next few months, it's very possible there may be no moratorium. And I think part of the problem that we have is that all of this trend, this revolution in genetic modification applying to humans, the public hasn't known about it. Nobody has been saying, look, this is a big deal. This is a revolution, and this could affect you in very personal ways. And so part of my goal is actually to change that and to educate and engage with the public and get you guys talking about this. And so I hope at these meetings that there'll be a role for the public, to bring their voice to bear as well. So if we kind of circle back now to 2030 again, that imagined story, and depending on the decisions we make again today, literally we don't have a lot of time in the next few months, the next year or so, because this technology is spreading like wildfire. Let's pretend we're back in that reality. We're at a park, and our kid is swinging on the swing. Is that kid a regular old kid, or did we decide to have a designer baby? And let's say we went the sort of traditional route, and there's our kid swinging on the swing, and frankly, they're kind of a mess. Their hair is all over the place like mine. They have a stuffy nose. They're not the best student in the world. They're adorable. You love them. But they're on the swing next to them. Their best friend is a GM kid. And the two of them are kind of swinging like this, and you can't help but compare them, right? And the GM kid is swinging higher. They look better. They're a better student. They don't have that stuffy nose you need to wipe. How is that going to make you feel? And what decision might you make next time? Thank you.
For that. It can be frightening to act, but sometimes not acting is worse.